Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled The Role of Chromatography and Monodisperse Particles in Mass Spectrometry-Based Metabolomics for Disease Detection. My name is Dr. Carrie Haslam, and I'll be moderating today's presentation. Today, I'm delighted to be joined by Dr. Timothy Garrett, Associate Professor at the University of Florida, as he discusses the clinical translation of metabolomics using examples of different diseases, including rare diseases, cancer, and infectious diseases. Timothy will also explain how critical chromatography is to reproducibility and metabolite identification, and how new monodisperse columns can aid in separation and identification. After the presentation, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your questions to the left of your screen. So without further delay, I'd like to hand over to Timothy for today's presentation, and I'd like to thank him for presenting with us today. Please go ahead.
Well, thank you for that wonderful introduction, and it's wonderful to be here to talk about the role of chromatography and monodispersed particles in mass spectrometry-based metabolomics for disease detection. So today I'm going to give you a short outline of our center at the University of Florida called the Southeast Center for Integrated Metabolomics. I'll then move into talking about the omics and specifically talking about metabolomics. I'll give you an introduction into monodisperse particle columns and some examples of how they're separating small molecule isomers. And then I'll talk about a couple of different disease examples. One primarily I'll talk about is meningioma and how we used a multi-omic approach to try to identify biomarkers. I am director of a research center called the Southeast Center for Integrated Metabolomics. We provide tools and resources to conduct metabolomics and lipidomics within the scientific community, both academic and industry. We focus on thinking about technology and how technology can impart a significant effect on the way we do analyses. We also focus a lot on building tools that are open source so that the access to doing good analyses is not prohibitively expensive. We don't currently have a granting program, but we will seek opportunities to give people collaborative chances to work with us as we continue to provide resources to the community. And one of the things I, of course, love to do is train scientists, graduate students, postdocs, and undergraduates in the art of metabolomic science and the technologies that are involved in this science. We are located here in the University of Florida and in Gainesville, Florida. And we have worked with many different people across the United States. This is just a scattering of the different locations where we've received samples. And we've also received samples from overseas and different research studies. We have several different methods that are available within the center, and I also run a research lab where we do other kind of technologies and development. But within the center, we run targeted quantitative assays, including those that are listed here. We also run customized method development if you have an analyte of interest that you'd like to develop. And of course, we run untargeted metabolomics using two different separation approaches, both Hillick and reverse phase, and lipidomics using reverse phase only. We also have a lot of experience in bioinformatics, which includes classic univariate and multivariate, as well as machine learning approaches to help identify markers of interest, as well as to help identify metabolites that are significantly different between your studies. And then we can have education programs and training programs for individuals who are interested in conducting more of this work. We are, of course, in the era of omics and really multi-omics, where we have a lot of it started with the genome and understanding what is involved in our genetic code. That can really describe what can happen, not necessarily what's happening, but the possibility. The transcriptome is what is then engaged, what is starting to turn on, which then leads to proteins changing or enzymes changing that are making other things happen. And those proteins and enzymes work directly with small molecules to have one input and one output, or multiple inputs and multiple outputs of those enzymatic reactions. And when we talk about metabolomics, we talk about two basic names. One is the metabolome, and that is really the collection of these small molecules that are present in tissue, plasma, urine, whatever biological fluid or plant-based sample or water-based material that you're working with. And then we use the term metabolomics to describe how we measure those metabolites. And ultimately, metabolomics is the comprehensive characterization of small molecules in any given biological system to gain an understanding of what may be happening under a certain stimulus or a certain event. In humans, this can be very complicated because we have many different factors that lead to our individual phenotype. We, of course, as I talked about earlier, we have our genes that start the processes of metabolic change. As we age, our metabolites will change over time, depending on how old we are and depending on other aspects such as where we live. And if we live in a highly polluted environment that has a lot of gases in the atmosphere, that might alter the kinds of metabolites that you would detect inside your body. If you are sick, whether that's a chronic illness or an acute illness, that of course will be a change in your metabolome, and that's one of the areas of interest that I care about, is understanding how we can find biomarkers of those diseases. If you take an over-the-counter medication or a prescribed medication, those will show up in your metabolome. They may not be important in terms of what's biologically relevant, but they'll show up and they're part of your phenotype. And likewise, your nutrition, whether it's short-term or long-term changes in nutrition, will have a biological effect that we can measure. And then exercise induced changes or sleep lifestyle, all those factors will affect what kind of metabolites you might measure, as well as when you take a sample, whether you take a sample at night or in the morning, that might have an effect on what metabolites you would see. So we are very diverse, both in who we are as individuals, but also in the metabolites in which we measure within our body. Put it in a little simple schematic, we have all of these factors that are at play within our biological system, and we're trying to use a analytical approach to separate and measure what's present. And primarily, we care about this part, which includes metabolites and lipids. And this is just an example of looking at the separation and measurement of tyrosine from this complex mixture. Ultimately, the goal of metabolomics is to define each one of these extracted ion chromatograms as a unique feature, which with a value that looks different or is the same between different sample types. So here's an example of phenylalanine that's eluting at 6.25 minutes. We have an extracted ion chromatogram for that. We have a mass spectrum that defines what's hidden under that and what masses are measured. And so we really identify a metabolite based on its mass to charge value and its separation capacity. And that's a key factor because the more ways in which you have to identify a metabolite, the more confident you are in its match. And so we would run a standard of phenylalanine in this case to make sure it matches retention time and the mass accuracy. And that would give us both a retention time and a full scan mass match. If you did tandem mass spectrometry, that would give you another factor of identification confidence to make sure it is really true. Note that you can also search against a metabolite library, but if you're searching against a library, you're only using one form of identification, and that's not enough to identify the metabolite with high confidence. And in the end, what the goal is to run several samples across different study section groups or different kinds of stimuluses that are going on to align all those features across those different chromatographic runs and create a aligned database that shows the mass and retention time, and then use that database to then do statistical analyses to identify what's unique and novel between those two groups or three groups or four groups. And then you would repeat this experiment again to make sure that what you get through statistical analysis is indeed correct. And then you have this extra confidence in the statistical analysis approach. So it's a combination of chromatography and mass that makes a very big difference in how we do metabolomics and how we conduct an identification process in metabolomics. And so that gets me into part of what I'd like to talk to you about today in terms of science is the evaluation of monodisperse particles and their potential in improving how we do metabolomics across a large subset of samples, or improving how we separate metabolites using this new approach in this new form of particle column. So a brief background, we are very common with the columns that are on the market, which are polydisperse. You may or may not be aware of that, but they have a particle dimensions that range across the desired average. So if you're 2 microns, you're going to have a particle dimensions that vary plus or minus a certain percentage based on how those are manufactured. In a monodisperse case, you're trying and creating a process in which the particles are very tight and closely spaced around the particle dimensions that you've defined, and so you have a much more uniform particle packing and much more uniform particles within the system. And so this is just an example of poly versus monodisperse. Would be an example of what that equates to in sort of the tightness around the particle size. So if we look at this example that we have with 3 micron particle size, we have a much less distribution across the particle dimensions, so a tighter distribution, and then you have 2 commercial particle columns where you can see that much broader distribution across the particle size, where one might have a smaller average particle size, but has an overall higher polydispersity. And so one of the things that this makes a big difference in is this factor between particles, this eddy diffusion factor, where if you have polydispersed particles that are the very non-uniform distribution, you're going to have a much bigger diffusion range within the particle, and that's going to affect peak broadening and band broadening. And if you look at this as an example, you see on the bottom case where we have a polydispersed particle column, and you can see this distribution where we have a very circuitous route through the column, where it's not very well defined because you have a lot of different interactions between the particles that are one size versus another size packed within that column. Versus in a monodispersed particle column, you can see how that tighter packing creates a much more uniform uh, movement through the column, right? And note that this is not a single analyte. This is a packet of analytes, right? So a packet of analytes is really what you care about in terms of that separation. So on this polydispersed column, you'll have a little bit more peak broadening because each particle, each analyte that's going through that will have a slightly different mean free path. And in the monodispersed case, you're going to have this much tighter peaks that are produced because they all have a more uniform path and they all follow a very similar path through that column. And so what I'm going to talk to you a little bit about is a test case when we started looking at a C18 PFP column, 3 micron particle size, monodispersity with 2.1 millimeter ID, 100 millimeter length. This is the classic phase and length of column that we like to use for metabolomics because it's a good mix between reproducibility, ruggedness, as well as the separation of polar and non-polar analytes in the same given run. And so this is an example of one method that we have that we've been using for a very long time in terms of the gradient and the phase separation. And so we primarily have been using C18 2 micron particles that are polydispersed for the past 5 or 6 years. And so we take this same gradient, same method, and you can see the gradient details here of what we're working with. And this is just a simple formic acid and water in A and acetonitrile in B gradient with a 25 degrees Celsius temperature and a simple 2 microliter injection. Note that this is a plasma extract where we're doing protein precipitation to prepare that plasma extract for metabolomics. And these highlighted arrows are some of the metabolites I'll discuss in the next few slides. So the first one is looking at 2 isomers of 5-aminopentanoic acid and valine, and this is part of the aqueous portion. So we have a linear, sorry, not linear, a isocratic portion from 0 to 3 minutes. So this is that isocratic portion where we're 100% aqueous. And note that these isomers have very different retention, which is excellent, right? And so we have valine, which is dilutes later versus 5-aminopentanoic acid. We have valine, which is dilutes later versus 5-aminopentanoic acid, which dilutes roughly about 0.8 minutes. Note that the void volume in this is probably close to around 0.5 minutes. So we get this increased retention time of polar analytes for at this phase of the column. We look at another set of polar analytes, isoleucine and leucine, 2 isomer amino acids. And we see the change here is a change in a methyl group. Note that this change here and here for leucine and isoleucine, and we get a good peak separation between the 2 isomers as well as excellent retention. And again, this is the start of the gradient at this point, so they're just starting to get eluted off this example. And you can see the full width at half max for ETH is nice and tight. Another one, and is an example that we have is acetylcarnitine, which is a common metabolite that we measure in metabolomics. So here's an example of preformed positive charge, so easily ionizable, and also would have a very different retention and characteristics in terms of separation. And note here we have a very optimal peak shape, a very Gaussian defined peak, and excellent retention for this polar analyte. And the last example before I show you comparisons between two columns is tyrosine versus phenylalanine, which is really just the change of a simple OH group on the phenyl ring for phenylalanine and tyrosine. And note that you get this extra long retention time for phenylalanine with that change in tyrosine. And of course, they have different characteristics in terms of their peak shape, and overall sensitivity is dependent on the concentration in plasma because they're not at equimolar concentration. And so then let's look at some differences between a column that we might use in a versus that we've been using for a long time versus a column that we're planning to move to, which is this monodisperse column. And so here's an example of the method we've been using for many years. It's the C18 PFP column, 2 micron, and it's polydisperse versus C18 PFP 3 micron and monodisperse, right? So we have a little bit bigger particle size, but note that we have this excellent improved retention based on the monodispersity, most likely because we have more interactions with the particles, and we can separate those analytes across that chromatographic range. Note we also have very similar retention difference between the two peaks for both. So baseline separation, which is excellent, that allows us to continue to separate isoleucine and leucine from each other. We look at that example of valine and 5-amino pentanoic acid. We note that in the case of the polydisperse versus monodisperse, 5-amino pentanoic acid has the very similar retention time, but valine is retained much longer. And note that we have a much improved Gaussian peak shape with this monodisperse column versus polydispersity. Likewise, you can sort of see that cleaner here with 5-amino pentanoic acid, and note in red is the monodisperse column. The next example is looking at acetylcarnitine, and this really highlights the improved peak shape between the polydispersed particle versus monodisperse. And note that in this case, we have an earlier elution of acetylcarnitine versus the classic polydispersed column, but the improved peak shape increases our intensity because now we're instead of broadening, getting peak broadening, which would reduce our sensitivity, we get this tight peak that allows us to be much more sensitive and for measurement of acetylcarnitine. And this last example, I'll show you another one after this that shows the same analytes, but in a different kinds of sample. So this is again a plasma-based sample looking at 3 micron versus 1.7 micron of the same column, PFP, C18 PFP. So just reducing that peak shape, just reducing that particle size down, and now note that now we have a little bit longer retention of leucine and isoleucine, and a little bit better baseline separation between the two peaks. And there's also this peak that, and if you had been noticing this peak that shows up as a little blip early on, it shows up a little bit better here. And if you look in the next slide, you'll see a little bit more what we think this peak probably is. And this is showing an example of 3 different columns. So the C18 PFP at 1.7 polydisperse and the C18 PFP at 2 micron polydisperse versus this monodisperse column at 1.7 micron. And so this is a fecal example, so a fecal extract where you have a much more complex mixture of metabolites due to the activity of the microbiome and the metabolites that they produce and impart into our system. And note here we have this same little blip that we saw earlier, but with this type of sample, we note that this peak is showing up at a much higher rate. And we know that this is either allo-isoleucine or nor-isoleucine based on running standards, but we cannot separate allo or nor from each other. So we have this introduction of this new isomer because of just using a monodispersed particle and a 1.7 micron distribution. Also note that alanine betaine is identified here, and this was identified by MSMS. So we note that we can identify that very cleanly. And there's another smaller peak earlier, and note that the retention time is a little bit longer in this case. And what this really equates to is when you do analyses and integrating peak heights, you can separate nor-leucine or allo-isoleucine much better. Now, in this case of a knockout, and this is just knocking out a specific micro versus wild type, we don't see a very large difference in that peak, but because we can separate them, that would allow us to look at other factors that might affect identification, and the biological factors that affect how norleucine or alloisoleucine would be created or metabolized. And note, we can also get a clean separation again in analysis of leucine and isoleucine, and the clean difference between alanine betaine, which is highly elevated in this knockout model, which allows us to then accurately predict that it will be a key biomarker for whatever this knockout model is assigned to. Okay, so hopefully I've showed you a little bit about polydispersity versus monodispersity and the advantages of using monodispersity in doing biomarker discovery and metabolomics. And in the next part of this talk, I'm going to give you an introduction into an example of how we use metabolomics to identify biomarkers of a disease. So this is a study of meningioma, where we're using both metabolomics and lipidomics, as well as artificial intelligence approaches, um, to identify markers. And note that this was actually just published a couple days ago in the Journal of American Society for Mass Spectrometry, which is a which is a great anticipated publication of this article. Meningioma is typically considered a non-cancerous tumor that arises within the membrane surrounding our brain and spinal cords. It actually accounts for a fairly large percentage of tumors, and that's around 37% of those in the United States. And they have three grades in which they're classified. Grade one would be a low reoccurrence and not high risk. So it's really just a removal, and then follow up to make sure everything's okay. And then you have the differences then now become higher grade at grade two and grade three, where grade three, of course, is the most severe, which has a very low survival rate and a high frequency of reoccurrence. And so our goal was to try to investigate the metabolomic profile of tissue types extracted from meningiomas, from brain samples from humans. We noted that we did not have enough samples in grade two and grade three, but one of the goals of the study was to try to see whether or not you could classify one versus two versus three. And so the primary goal, or our initial goal, was really characterizing low grade tumors versus high grade tumors, with the ultimate goal is identifying biomarkers that could differentiate stages of meningioma and provide a better diagnostic test for clinical pathology. And note that this is a excellent collaboration that we have with anatomical pathology here at the University of Florida, where we have a very highly collaborative environment to be able to conduct research like this. So this is a little bit complicated, but not that. When you break it down, we basically have two parts of what we wanted to do here. One, we wanted to do a classical experimental analysis where we get the resected tumors, we pull out a sample, we extract those for lipids and metabolites. So we actually use two methods. We use the Fulch method for lipids, and we use a protein precipitation method for metabolites. They were all conducted on a thermo Q-exactive with a Dionex UHPLC, and metabolomics was conducted with C18 PFP column. And then we did statistical analysis in both MetaboAnalyst as well as scripts that were written in Python. In the second phase of this, we combined all the data together that was generated from whether it was metabolites or lipids. And we then looked for feature selection using this dimensionality and reducing the data based on the significance of features, that then fed into machine learning models to continually reiterate that process to see if we could really find very significant and important metabolites. And so if we look at the example of just looking at metabolites and lipids, this is an example of lipid separation and using hierarchical clustering analysis. We've classified both 2 and 3 as together in order to test the machine learning models first, as a way to see whether or not, yes, indeed, we could separate out grade 1 versus grade 2. And here, note one of the largest differences is an identification of triglycerides. Triglycerides are elevated primarily in those grade 2 and grade 3 tumors, and you can see this dot plot that shows really the distribution is primarily towards saturated, not unsaturated, fatty acids with longer carbon chains. So you have a increase of the region in this dot plot of long chain carbons with very highly saturated fatty acid chains. And This is equated to understanding growth for cancer. In many cases, cancer cells obtain fatty acids from lipogenesis, but they need to turn over very quickly. So they're really getting these saturated fatty acids as a part of that process to grow very rapidly. And so this could be a way in which we're monitoring the process of how these are growing. And note that the distribution here is primarily from 16 to 24 and even up to 26 carbons long within that region. And so we also look now at metabolomics, since we did these as a separate analyses, and we can sort of dig through this metabolic pathway, right? And we can see that we have changes in pyrimidine metabolites where they're lower in grade 1 tumors and higher in grade 2 tumors. So we have an increase in, say, nucleotide synthesis. And the same goes for purine metabolism, where we see this increase in nucleotide synthesis for the processes of growth in grade 2. Um, Vitamin B6 is a metabolism primarily looking at pyridoxal phosphate and pyridoxate, as well as riboflavin, are downregulated in grade 2 tumors. And we don't necessarily know exactly whether that's a nutrient that they're pulling from the environment, or if it's a result of just changes in the cellular environment where there's less vitamin B's available to be metabolized. Or it could also be related to the gut microbiome in some ways, because we know that we get many of our vitamin B's from the gut microbiome. So there could be factors that are more complex than just understanding the tumor environment. But do remember that these are just tumor tissue samples, not plasma. So we're looking directly at what's sort of happening in the tumor. So it's probably less likely a microbiome issue and more of a nutrient reduction issue. And so then we take a step back with this study, and we could look primarily at those together and individually and see what's going on. But we wanted to see what we could do with machine learning or artificial intelligence approaches with this kind of data set. What we know in metabolomics is that we create a large type of data, especially when we do multi-omic studies. And this is just lipids and metabolites. But if we layered in proteins and we layered in genomics, we would have many different omics approaches. You could even layer in glycomics. And so you're dealing with a large data set, and it's nonlinear. You have very high abundant signals and very low abundant signals. You have polar and non-polar metabolites that are mixed together. You have preformed positive charges versus not. So it's very heterogeneous in what's measured. And we've seen over at least this data is really just up until 2020, where we see a much huge growth in the application of machine learning in metabolomics. And if we had 21 and 22 data for this, I would assume that this also follows that same exponential growth, mainly because there's a lot of tools now that are available to conduct artificial intelligence, and of course, a lot of interest to be able to build these kinds of tools, especially with Chat GPT and those kinds of artificial intelligence approaches that have been made more commonplace. So really what's happening in machine learning is it's just a process of teaching a computer how to make predictions, right? And so we have ourselves at the front of this. We provide data to it. We do some data cleaning up because we know how to clean up the data. The software doesn't necessarily know what that data looks like, right? And then we build a model based on that initial data. Um, and we go all the way to the end, right, to this model testing point, and then we continue this loop, right, to build and train the model. And then we would go back from this test and provide a test data, right? So you would split your data up. Part of the data would be made to build the model. The other part of the data would be used to test that data into this model that you've built. So you sort of keep this group of data away from the model initially, and then add it back into the model to make sure that the model you're building is actually precise and have a way to test it. A key part of this is you can't use small sample numbers. If you're trying to do machine learning on a small study set of, say, three samples per group, it's not enough data to really be able to test whether a model is sufficient. And so we're just under 100, around 86 samples. And that gives us enough to be able to split the data in half. And we can do this model testing. And then eventually you would deploy a model, and you'd start with a new set of samples and go through this whole process. And note that this is used all over the place, right? Face detection, handwriting, healthcare, of course, is a large growth area of interest for this. And so what does it look like when we do this? Well, we had initially started with 17,000 features. That's both lipids, metabolites, positive and negative ionization data sets. So 4 data sets that were compiled and put together in one large data set. We started with this Python scikit learning package, which is a common approach in the Python tools that are available. We use the extra trees package to do classification and regression. And then we had this list of feature importance that comes out. And so we labeled these as a lipid that was from positive ion mode with its mass and its retention time. And likewise, metabolite that was positive mass and retention time. And in other cases where we knew and confident in the ID, we labeled that as the metabolite or lipid of interest. So in this case, I'm showing inosine and 3' CMP. So this is just a list of feature importance, but we have to filter that list. So this is all 17,000 features, right? And so you can sort of rank this list on those that are significant based on this extra trees package. And then you can pull out only those that are highly significant that are really starting to make this inflection point. And this gets us down to really 30 or so metabolites and lipids within this window of data. And then we feed those 30 within this model to a machine learning algorithm where we're now testing 6 different machine learning approaches to see which one might have the most predictive behavior based on our model and test data. So we're feeding both our model and test data into this, and you can sort of see how they work based on the number of features. Ideally, more features often gives you a better predictive algorithm, but not always the case, right? But in almost all cases, except for the decision tree, we see as we increase the number of features, we have a much more accuracy in the prediction of what the types of samples are. And we thought right here around 90% was a good 90% accurate was a good place to go. And that includes about 5 unknown metabolites within that list that we could spend our time. And note that support vector machine learning was the most precise model in building this. And so then our um, this is a list of those metabolites. You can also see them over here to the left. We had two lipids, one from positive, one from negative ion mode, and one metabolite that was identified in positive ion mode. We can then go back to uh, the box plots, and we can indeed confirm that yes, this metabolite is elevated in this feature number 1, which was a lipid. And feature number 2 was another lipid. Both are elevated. And if we look at number 3, feature number 3 was also elevated in grade 2 and grade 3. So we have 3 elevated metabolites and lipids that we could use to potentially identify what new biomarkers might be. And so this is really the complicated part of metabolomics, right? We think we can identify metabolites, but it's probably one of the more complex ways that we have in our world is trying to figure out how to identify true metabolites that are unknown that we don't have any information about, right? So we have several ways in which we can do that, right? The mass spectrometer gives us exact mass. Retention time gives us some characteristics that help us understand how the polarity of the analyte. We can calculate a molecular formula. We can get a tentative structure of that. Um, and then we can potentially get a putative structure, but mass spec can't give you a full structure, right? And then level 1 is a fully validated structure, right? And so we have databases that you can search to get hits. We have MSMS data that we can use to help identify structure based on fragmentation. And so in this case, we had 5 in the list. We were only able to identify 3 of the top 5, and these are the top 3 that we were able to identify. 2 of the top 3 were lipids. One was a triglyceride, which made sense. It fell in line with our preliminary analysis of looking at just the lipids by themselves. And it also falls in line with the saturated and long chain carbons that we saw in that initial analysis, where we have 2 long chain fully saturated fatty acyl chains and 2 degrees of saturation for a triglyceride that was identified. We also identified a galactosyl ceramide as part of the process, also following in similar line with this high degree of saturation and long carbon length. The final one we had was a peptide, which was unusual because most of the time we think we're not measuring peptides in metabolomics, but you'd be surprised. Many things that you might identify are peptides that make it through the entire process of extraction. Note that we did do a protein precipitation process, right? So protein precipitation will remove proteins, but not necessarily small peptides, right? And so this is a 14 amino acid peptide sequence. We have identified the full sequence, but we don't yet know what protein that sequence is derived from. And so what we were able to do as a summary of this meningioma study is we were able to use machine learning with a combined data set of metabolites and lipids to identify 5 unknowns that were highly significant in grading tumors based on 1 versus 2 and 3 at a 90% precision rate. We were able to identify only 3 of those and 2 still remained unknown. We did not have enough data to be able to separate and do classification of 2 versus 3, primarily because we didn't have enough in grade 3. We only had 15 of that group, and that was not enough to really truly rely on machine learning. And so the goal of that would be to have more samples and build a larger database to then be able to differentiate grade 2 versus grade 3. And of course, we have this understanding the biology of this takes a lot of time, right? We know that lipids are harnessed by cancer cells to grow. They have to have their own lipids. They hijack those lipids from this content of lipid droplets often to continue their process of growth. And that long chain carbon could be important um, in terms of saturated levels and targeting that maybe in the future. But we don't. A lot of that would have to be done in other work to try to understand more about the biology of these changes. So then I'm going to switch gears here for the last few minutes of this lecture and talk to you a little bit about dried blood spot metabolomics and a couple of applications and some innovation that we hope will help make dried blood spots more reliable. Note that dried blood spots are probably one of the oldest forms of collection for metabolomics that we know about, where mass spectrometry started with phenylalanine in the 1990s. Much prior to that, we also started using dried blood spots for doing PKU analysis without mass spec. And so PKU analysis had been done for a very significant amount of time, but it was the introduction of electrospray as well as the introduction of the triple quad that made dried blood spot analysis for phenylalanine and then also all of newborn screening much more reliable and useful in the detection of newborn errors in metabolism. And so this extraction is and derivation is very simple. It uses a simple methanol or acetonitrile extraction followed by butyl ester derivation for classic analysis with electrospray ionization for high sensitivity and automation connected to a triple quad. It's often using section of scans like neutral lock scanning or precursor ion scanning, as well as more recently selected reaction monitoring to look at a more lower level and lower abundant metabolites. And in standard dried blood spot analysis, there's some imprecision because you don't necessarily know the volume of blood that you're using on a small spot, and the hematocrit levels are vary over time. Some applications I already talked about newborn screening, which is the most common application of metabolomics, clinical metabolomics, as well as the most common application of dried blood spot analyses, where in the US we have over 4 million metabolic screens done every year. Other areas that are commonly used are often in toxicology for measuring blood spots either in remote locations or collecting those samples, as well as infectious diseases, where you can transport blood spots on cards that are from patients that might have been infected, and on transport on dried blood spot cards is considered not to be an infectious material because it is dried. But you can also use it in therapeutic drug monitoring, where potential home sampling, where you're collecting samples and a patient would collect a sample to monitor their concentration of their drug in their body. And home sampling, of course, became a much more common uh, opportunity with COVID and with lockdowns, where we wanted to provide patients opportunities to get tested, but also prevent transmission of viruses. And so this led to an increased need for home sampling tests, of which dried blood spots is one of those possibilities for. There's also a need and desire to use multiomics of using dried samples because liquid samples can be difficult to transport. They cost a lot to transport samples across the United States, or of course internationally is very expensive. And so you can collect dried blood spots in remote locations and ship via easier mail approach versus having to ship on dry ice for liquids, and then also using a -80 freezer for liquid storage. Also using a -80 freezer for liquid storage. And so the need for small sample size is important, that are stable and reliable. So we had a study that we conducted in malaria with a collaborator here at the University of Florida, Dr. Julie Moore, where the goal of this was to look at malaria and pregnancy. We know that when a mom gets pregnant, the malaria affects both the mom and the infant that's inside. And we also know that pregnant women have a higher risk of infection, especially the first pregnancy. The premagravid women are much higher risk for adverse outcomes. All right. So this includes issues with low birth weight in the infant, reoccurrence rate because of reinfection within the system, and poor developmental behaviors. And so, preventing infection or understanding infection from a more biological meaning would be very important. And so, this is a study that Dr. Moore has been involved with for a very long time in Kenya. They recruited over 6 years, roughly 1,000 patients. With clinical data, placental blood, as well as whole blood, peripheral blood, and tissue samples that were collected in remote locations that are difficult to use. So this is a very important cohort because it has a very good clinical measure of what's happening with positive patients, low birth weight infants, as well as those that have HIV. So note that this is a complex kind of study in a complex area where we have multi disease that happens, including both that are malaria positive and the PM plus, as well as HIV positive. And so locating uh, uh, mothers that are in their first pregnancy it can be challenging in this setting. So this cohort represents a very unique kind of sample set where we have a large number of patients. We note that in many cases the placenta manages to support a very successful pregnancy most of the time. But there's also issues when it's not supported. What happens? Why, when it's not successful? We'd like to know more about the biology of that. So here's an example of the kind of cards that we would collect. Those cards would be from blood spots like this. So this would be an example of one that's spotted from, say, placental blood, or one from peripheral blood that's collected and dropped onto there. And then you have this dip card where it's dipped in the sample and then stored in these cards in this kind of way and be able to ship in this mode. And so our study, we were able to get samples for this study to look at metabolomics and lipidomics across this study group. The samples we have were cut out, and we extracted both lipids and metabolites using the Fulch method, which is a combination of chloroform and methanol that allows you to separate the both lipids and metabolites from the same sample. So it utilizes one blood spot card and allows you to get both types of samples from that card. We then conducted analyses on a thermo-Q exactive using reverse phase separation for lipids as well as for metabolites. And we used an approach where we can dig into the data called iterative exclusion tandem mass spectrometry, which allows us to increase coverage for lipids as well as metabolites. And in this case, we wanted to compare the expression of lipids across both malaria positive as well as malaria control samples, so no malaria. And so here's a case of looking at P falciparum positive versus negative. And this is an example of only 4 patient samples that we had across this group looking at that biological separation. It's clear that we have a clean separation between those that are positive versus those that are negative. And so this pilot study allows us to at least validate that we can two things. One, we can use blood spot cards in malaria research collected in remote locations and collected over 6 years ago to show the feasibility of using blood spots to identify potential differences between patients. And if we look at some of the expression across this, we see the heat map clustering of the top 50 or so metabolites and lipids that are present. We note that we have clear clustering associated with ceramides that are elevated in malaria positive patients, as well as phospholipids such as PC and PE that are elevated. And then we also note this extensive decrease in cholesterol esters that are in the positive P falciparum patients. Um, this decrease in cholesterol esters could be related to an increase in cholesterol in the in the malaria growth or development. We don't know yet, but because we know that parasites need cholesterol for growth and replication. But in this case, we don't necessarily have isolated malaria parasites. We just have the blood, and that blood at least shows us that this reduction in cholesterol esters correlates to a high positivity rate of P. falciparum. And we know also that this type of malaria lacks the ability to synthesize cholesterol. So it has to grow, it has to take it from its environment. And so there is biological meaning behind reducing that within in the environment. And this is really just a pilot study, right? It's only 4 samples, a way to sort of us to quickly test whether or not we can use old samples and that whether they would be useful for doing biological interpretation. And yes, in this case, we do. And so now we're undertaking a much larger study through roughly 100 subjects and looking across different disease infections, both HIV and malaria, to try to understand more about what could be happening. But this also allowed us to sort of revisit the concept of a dried blood spot, right? And so we wanted to try to improve how we do dried blood spot analysis. And so we started working with a company called Capitainer that makes this very precise blood spot that gives you exactly 10 microliters of collected blood. And so this is just showing a comparison of a classic Guthrie card or Wattman 903 card, and showing the size dimension of what this blood spot would look like. And so its size fits perfectly into 96 well tray. So you can actually punch it out and put it into well, you don't have to punch it out because it's exactly the right size. You just drop it into the 96 well tray for extraction. And this is what the card looks like. It's a quantitative dried blood spot card because it quantitatively delivers 10 microliters of blood from a finger stick. So you would put your blood sample here. Underneath here is a little capillary that draws the blood over to this spot and transfers only 10 microliters, with the rest going to a waste area. And then you have a dried blood spot card or a dried blood spot here that you could use for doing different kinds of analyses. And so what we also wanted to look at is how we could improve quantitation and analysis of small molecules, right? So in a classic case, we have a blood spot card that's, let's say, has 200 micromoles of material, analyte in it. We go through the extraction process. We assume that we get 200 microliters out of the blood spot and that we have 100% recovery, but in reality, we often, and we will, lose a certain percentage of analytes into the card because they cannot be extracted, whether they're stuck in the card or whether the extraction solvent isn't optimal. And that would equate to some variable recovery. I'm showing 90%, but it could be 60, it could be 40. We don't know, because if you're not measuring recovery, you won't know how much is lost. And so if you had an internal standard present early on, you could account for that loss, right? So even if you lose 20 or 50 micromoles of the initial material, you're going to lose a similar amount of the internal standard, that's labeled version of that analyte, to give you essentially 100% recovery, that would be measurable based on the reference of the analyte to its internal standard. And so this is what we did. We developed a alternative to the quantitative dried blood spot, where we add an internal standard to the card prior to blood collection. So it sort of standardizes the blood collection at the time in which you collect and transfer the blood onto it. And so in the classic case, you have a few drops of blood that are put on, you punch out this blood spot card, and then you add your solvent with internal standard. But in this case, since the card is already contains the internal standard, all you're doing is adding solvent and extracting. And now you're extracting the internal standard and the analyte at the same time. And then an example of this would show amino acid profile of both the target analytes as well as the respective internal standards that would be extracted at this time. And so you eliminate some of the error because you're not making internal standard anymore. All you're doing is adding a solvent to extract. And then you can sort of see an example of this, where 3 different cases where you have in case A, you have the blood and the internal standard is in the card, versus B, where we're adding it into the plasma and then adding it to the card, and then C, where we're adding it only in the solvent for extraction. And so in the solvent, you get 100% of your analyte back, but you actually get an incorrect ratio because it's not accounting for the extraction efficiency of the card. So this is reality, and this is what we think this is what really equates to reality. This is what we're currently doing right now. So you see a difference in terms of that extraction efficiency that we're not measuring if you don't have the internal standard in the card. And that then equates to higher reproducibility, and then also equates to better quantitative analysis. And so this is just showing calibration curves where you're adding the internal standard, and now you're getting a much more reliable quantitative value. And we have a lot more future work to do with this, primarily starting with larger patient cohort, looking at multiomics with this kind of approach, and then collecting samples in remote locations to look at this internal standardization process. So the internal standard in the card mixes with blood at the time of collection, which is very precise and it allows you to count for extraction efficiency. It also accounts for potential degradation, because the degradation would occur to the internal standard as well. And you could add multiple isotopic internal standards to this, and we've tested up to 500 internal standards using a Roa yeast extract for this. And then we can simplify the lab process on the back end because everything's contained in the card. And so with that, I'd like to thank you for your attention. I'd like to thank the individuals involved in a lot of this work. The malaria work was done by Sina in my lab, and the dried blood spot quantitative analysis was done by Wenchen here in the lab. And then I had a significant amount of collaborators. Another former student Hoda, who did most of the work in meningioma, and then collaborators in clinical pathology that have assisted, and as well as the funding that has allowed this to occur. So thank you very much for your time.
Thank you for that interesting webinar, Timothy. So let's move on to the last part of today's webinar, which is the question and answer session. So the first question we've got here is, why do you think the monodisperse column showed better peak shape for the metabolites shown, especially acetyl-carnitine?
Yeah, thank you for that question. I think one of the main aspects of that is we're reducing that diffusion aspect between particles by having less space between them because you have a more packed surface, and that reduces their interaction between particles, and so that allows you to have a much more cleaner interaction within the particles. So that gives you a clean response. And with the acetylcarnitine, which we've had issues with in the past, it allows you to get that very Gaussian distribution and increase the overall sensitivity for it.
That's perfect. Thank you. On to the next question. And this one is, why do you think you observed longer retention times for most of the metabolites using the monodisperse column?
Yeah, I don't necessarily know exact evidence for this, but I think it's partly because you have less of that gaps, less spaces between particles. You have more effective particles in the column, and so you have more interactions that those analytes will have with the particle phases, and so you're going to extend that retention time, especially for really polar analytes that have maybe less interactions, and you're adding additional um, retention against particles that are more tightly packed.
Perfect, thank you. The next question here is, in your meningioma study, you identified 3 out of the 5 most significant metabolites and lipids. What about the other 2?
Yeah, it's a really good question because we got down to the top 5, and those other 2 were metabolites, and we spent probably 6 months trying to identify them, and we're still not able to add true identities to them. And so it shows some of the difficulties when you have a true unknown, how hard it can be to identify them. Lipids have well-known fragmentation pathways, and so we have good knowledge in terms of how we can classify them. Same thing for peptides, which is what the other one was. And so we have 2 lipids, 1 peptide identified, and then 2 remaining unknowns that were real metabolites. And so the only real way to do it is to do fraction collection and then send those to NMR. But we don't have any remaining samples because these are biopsies from the brain, and so it's difficult to get new samples for that, right? And so we're still trying to figure out what other ways in which we could identify them.
Perfect, thank you. So following on from that study, what are the next steps for the study?
The next step for that one is to expand, to try to get more samples, to create a larger study, especially getting samples related to those that have um, grade 3 diagnoses, because we didn't have many in grade 3. And so it'd be nice to get more so we could start to potentially differentiate grade 2 and grade 3.
Perfect, thank you. We've got another question here, and this one is, have you tested any other analytes, that phenylalanine, sorry if I've not pronounced that right, and tyrosine for your quantitative dried blood spot approach?
Yeah, great question. We have tested other amino acids besides phenylalanine and tyrosine. We focused primarily on those just because they were a great way to validate it, and it's well known in terms of rare disease monitoring for PKU. But we've expanded now to acylcarnitines as well as all the amino acids, and so we have now good quantitative data on new analytes that hopefully in the next 3 to 6 months we'll be able to share.
That's great, thank you. Another question here is, the instrument gives 1 ppm accuracy. What ppm tolerance to use for data searching?
Yeah, good question. 1 ppm is achievable, but we typically use a 5 ppm tolerance to search, just to give a little bit of leeway if for some reason one scan in all of your mass scans falls out of that 1 ppm mass accuracy. And so that it's usually 5 ppm as a search.
That's perfect. Thanks so much. Well, that's all we've got time for today. So thank you very much to Timothy for that informative discussion and presentation. And thank you to everyone joining us online and for also sending in your questions. If you've got any other questions, please feel free to email me at editor@selectscience.net and I'll follow up with your questions after the event. If you would like to listen again to the webinar or maybe invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye and thank you once again for joining us.